Good afternoon. I'm Choi from State University of New York at Binghamton. Today I'm going to talk about direct visualization of electrogenic bacteria in a microfabricated microbial fuel cell. So this is all about energy harvesting from bacterial metabolism. For your better understanding about this research, let me spend some time in explaining some background. So due to depletion of fossil fuels and global climate changes, people started to look for the renewable and carbon neutral alternative energy sources. There are many types of renewable energy sources. You can use water, wind, or solar or geothermal energies. And also there's bioenergy. There are many ways to use bioenergy. For this research, we use the bacterial metabolism. So when bacteria break down the organic food, the NAD plus functions are repeatedly oxidized and reduced to generate the energy in the form of ATP. And for this reaction, they need electron acceptors. And normally oxygen is being used for this reaction. That's why we call this respiration. Now, if we engineer their environment so that the oxygen is limited, instead, if we provide a solid electrode, then we can capture the electrons and bacteria can survive. So we are using this phenomena to generate the energy using this microbial fuel cell or MFC. So this MFC is very similar to conventional chemical fuel cell, except that it does not need a catalyst in the anode. Instead, it uses bacteria. So bacteria generate the electrons, and also the protons must transfer through this ion exchange membrane to maintain the electron neutrality. And these protons are reduced at the cathode. So, but the problem is not every bacteria can do this. There are some special bacteria which have ability to transfer electrons extracellularly to the outside electrode. We call this bacteria electrogenic bacteria. There are many types of bacteria, electrogenic bacteria. These three species are the most popular right now, the Schwannella and Pseudomonas and Geobacter species. And those bacteria have their own electron transfer mechanism. And some of bacteria has this mechanism, direct transfer. And this type of bacteria has to contact with the solid surface. So the problem is bacteria that do not contact with the solid surface cannot provide electrons. So that's why this type of bacteria generates such a low current density. The second mechanism is shuttling transfer. So this type of bacteria uses some shuttling chemicals to transfer electrons. And the rest of the mechanism is nanowire transfer. And some of bacteria can provide conductive nanowires to transfer electrons to the surface. So as you can see, this type of bacteria can produce the best performance. So normally, these Geobacter species have nanowire transfer mechanism, and Pseudomonas has the shuttling transfer mechanism, and Schwannella has the both of mechanism. But the thing is, we are not sure when they prefer this mechanism or that mechanism. So when bacteria cells are introduced onto a solid surface, they tend to form a biofilm. And normally this biofilm formation is highly related to electricity generation. The thing is, we are not sure about the relationship between bacteria biofilm formation and the electron generation. So we need a technique for direct visualization of bacteria biofilm formation with the electrical measurement at the same time. So this is our project goal. So we developed a micro-sized transparent microbial fuel cell for direct observation of biofilm formation with electrical measurement at the same time. So as you can see, we have protruded the anode chamber, which is placed over the microscope. So we can see the biofilm formation with the direct observation, direct electrical measurement at the same time. So this is device configuration. So we used a PMMA acrylic substrate, and we used a very thin gold electrode. So the anode chamber size was about the 140 microliter, and for cathode, the 70 microliter. So we tested the two different bacteria species. One is Schwannella, and the other one is Geobacter. And I used the ferricyanide as a catholyte. So we do the direct observation and the electrical measurement at the same time. So this is the result of Schwannella species. So as you can see, they form the biofilm very quickly, within 15 hours. At the time, this is the electrical measurement. So open circuit voltage was about the 0.4. And when we connect the external resistor, there was a huge voltage drop, but it increases with the biofilm formation. So when they have the monolayer, the current was about the 5 nanoampere. And this result is about the Geobacter species. Unlike Schwannella, it took some time to form the biofilm. But as you can see, they form the biofilm based on these microcolonies. So this microcolonies becomes larger and larger. And here's another microcolony here. It becomes larger and larger, and they formed in a biofilm here. So also, likewise, the current increases as the biofilm formation proceeds. So for your better understanding, let me explain this. So this is a typical voltage of microbial fuel cell as a function of the current density. So at low current density, there was a huge voltage drop. So this is called an activation losses. And this activation losses is from bacteria electron transfer mechanism. So that's why I used the high value of resistor to study about this, the electron transfer mechanism. So let me compare these two bacteria species. So for Schwannella species, they had large activation losses. So here they had a large activation losses, which is about 0.42 voltage, while the Geobacter species has about the 0.2. And also, the Schwannella generated the 71 nanoampere per centimeter square, while the Geobacter species generated 4.8 microampere per centimeter square. So based on this result, while Geobacter species prefers this nanowire transfer mechanism, the Schwannella, for initial biofilm formation, this Schwannella may be used in this shuttering transfer mechanism rather than this nanowire transfer mechanism. But there are some challenges on this research. I admit that it was hard to see through over multi-layers. So when the biofilm form more than one layers, it was really hard to see through this thick biofilm. And also, this device had a large proton travel distance, which is about centimeter, while in typical micro-sized MFC had a micrometer proton travel distance. So normally this distance is related to the power density. That's why we got such a low power density and current density. So this is what we are working on right now. So let me conclude this work. So we built a transparent microfabricated microbial fuel cell. So visualizing electrogenic bacteria in situ with a real-time and simultaneous measurement of bacterial electron transfer. So thank you for your attention. Any questions?
Thank you. Unfortunately, again, we don't have